Hello, everybody. Um, my name is Peter Giuliano, and I'm the Executive Director of the Coffee Science Foundation and the Chief Research Officer of the Specialty Coffee Association. So thank you so much for joining us for this webinar presentation that we're calling New Brewing Research, Facts and Findings After 4 Years of Cutting Edge Coffee Science. And for this webinar, I'll be joined today with two colleagues, Phil McKnight from Breville and Professor William Ristenpart of the UC Davis Coffee Center. So I'll introduce both to you in a few moments, but first I wanted to do a little bit of backgrounding about this project and what it means and why we're doing it, why we did it, and some background. So first I want to start with the Coffee Science Foundation as an institution. We exist to achieve our mission, and our mission is this one: to advance the understanding of coffee and secure its future through research, knowledge building, and outreach. And for that, we get resources mostly from the specialty coffee industry, and we use those resources to increase the knowledge about coffee anywhere we can. And this project was one of our earliest and was begun 4 years ago, and I'll tell you a little bit about that. So why do coffee brewing research? And I'm talking here about filter coffee or drip coffee, whatever you call it, and which is a very popular drink, particularly here in the United States. And many of you are aware of some standards that we have around that, especially Coffee Association. But those standards that the SCA currently has were based on work and research done in the 1950s and 60s. Now, since that time, research tools have changed dramatically. You know, access to technology, instruments are less expensive, and the fields of both chemistry and sensory science, and particularly sensory science, have really advanced since then. So we knew we had the opportunity to bring brewing research into the 21st century by doing another round using modern tools instead of those that were available in the 50s and 60s. At the same time, consumer preferences have certainly changed over that time. So it was a perfect time to revisit some of these basic standards. So just at the moment that we were thinking about doing this, I like to say that two heroes appeared. And those two heroes were Breville and the UC Davis Coffee Center. Breville, 4 years ago, came to this SCA and said, look, we're a research-driven company, and we're interested in supporting some research around brewed coffee. And at the same time, the UC Davis Coffee Center, really Bill Ristenpart said, look, we're really interested in doing some coffee science in this field. And we realized that we had a dream team with these three organizations working together. So that started, Breville provided some guidance and a generous grant to support the work. UC Davis designed a research project, and that's what we started on 4 years ago. Since then, we've wound up with all of these outputs, 9 peer-reviewed academic publications that encompass chemical, sensory, and consumer research. This work has supported 9 researchers, including undergraduate researchers, postdoctoral researchers, doctoral researchers, and PhD professors like Professor Ristenpart. We've done 7 publications in SCA publications, and we've done dozens of lectures at various events that were all made available to the coffee community. Breville itself designed a tool that helped consumers optimize their coffee flavor that's based on this science. We're working on a new coffee brewing handbook that will be based on this science, a new coffee brewing control chart based on this science, and tons of other insights and data that you'll look at. So this is an amazing amount of output. And we're really grateful to this team for coming together to drive this output and provide all this knowledge for the coffee community. And this webinar is a little bit of a capstone or a reflection on those achievements. So we're going to try to get through as much of this information as we can with you on this webinar. So with that, I wanted to hand over the microphone to Phil McKnight, who, as I said, is from Breville, to get a little bit of his reflections on what inspired Breville to take this on and what their feelings are about this research. So, Phil?
Hi, Peter, Bill. Hi, everybody on the call. Firstly, I'd just like to thank everybody for the opportunity to just participate in this capstone webinar and review of all the research. I've got to say that it was nice to see Peter's summation of how the project started and then the outputs that I would say are just hugely impressive. Not only the number of peer-reviewed papers and work that's dropped out of it, but I think just the amount of support that the program was able to provide to the undergrads who were also working through the program as well, which is, I think, in its own right, very impressive. And I know that Breville is extremely happy and excited and very supportive of all of the work that the entire team has put together over the last 4 years. So yeah, about 4 years ago, Cliff Tong and Alex McKnight and I visited UC Davis, and we sat in a room with Professor Bill Ristenpart, Katie Goulding, Peter Giuliano, and then later we met with Jean-Xavier Guinard, and we talked about the project and what it could be. Originally, it started out as redefining the coffee brewing control chart, and I was quite excited about redefining it because I'd had to use that tool a lot over the years in presentations, and it was not easy to explain the way that brewing control chart worked to the uninitiated. So I was excited about redefining that work that had really gone back to the 50s and 60s and seeing what an updated version could look like to make it more accessible. But when we talked about other things that we could possibly achieve with the grant that we were providing, and there was some other ideas that we kicked around. One of them was the impact of brew water temperature on taste. I had some personal views and thoughts on that, which turned out to be totally busted in a very scientific way, which was it was not my ad hoc personal view. It was actually done with science, and that was a myth that was busted that I'd held on to for a long time. We also talked about the potential to test basket brewing geometry and its potential impact on taste and flavour. And Breville thought that we had a pretty good platform to test this because we had a brewer that had both a flatbed brew basket and a conical brew basket, and the water delivery and system was identical for both of them. So you're able to control temperature with a flow-through PID temperature control heater, and then flow was very consistent because it was driven by a pump. So we thought that with this as a platform, we could very easily and robustly test the differences between those two brewing basket geometries. So it was very exciting to see the output of that, and I've got to say that when we developed this product and we included those two brewing basket geometries in the development spec, we had no idea of its impact on taste or flavour. It was part of the product spec because it was our belief that in North America you needed flat baskets to satisfy consumer demand, and then in Europe and the Nordics you needed a conical basket to satisfy their brewing demand. So that was how we ended up with those two geometries in the single product, and we had we had no idea that it was going to have an actual, like a massive difference on taste impact for the same coffee with the same brewing parameters. So that's sort of where we came from and how we landed at Davis. And I've got to say that Breville's super happy and super excited about the work that the UC Davis team has done, led by Professor Bill Ristenpart and Professor Jean-Xavier Guinard. They did an amazing job garnering the teams and I suppose focusing the work on the tasks at hand, and then to have all of those outputs of peer-reviewed papers that have dropped out of this, I think has been an amazing effort. So congratulations, and we're very thankful and grateful to be involved in the research. So thanks very much.
Thanks so much, Phil, for those insights. I appreciate it. Okay, now I'll turn you over to the main event, I would say, which is Bill Ristenpart, and I think we can see it. There we go. We can see his screen now. So Bill Ristenpart is, of course, a professor of chemical engineering at the UC Davis Coffee Center. He needs no further introduction, really, because he's really, as I mentioned before, this there's a lot of this that was really Bill's brainchild. So I'll let him tell you all about it for the next hour or so, and then hopefully we have some time for some questions and answers at the end. So Bill, please take it away.
Awesome. So thank you so much, Peter. Thank you so much, Phil. It's really exciting for me to be here because I agree, this is in many sense kind of a capstone of many years of effort. And I'm really, really excited to be here and share these results with you today. And so what I'd like to first start with is very briefly, because my address is at the UC Davis Coffee Center. For those of you who might not be familiar with this, the Coffee Center is something that we created at UC Davis right around 2016 or so. And so this one slide right here summarizes in a snapshot of what we're at. We have a whole building here on campus at UC Davis. We have a pilot roastery, we have a brewing and espresso lab, we have a green bean storage lab. We've actually raised to date about $4 million in philanthropic contributions to renovate this building. It was already built, we're using it, but it's not really optimized for coffee. And so I've been spending a lot of time over the past several years raising funds, and the coffee industry has really stepped up. And so you can see here on the side, many of the entities that have donated very generously for the Coffee Center building. And I'm very happy to report that we had a site dedication ceremony just a couple months ago, where that's the chancellor right there. There's me, and we dramatically lifted the blanket off of the new sign for the Coffee Center building. And like I said, we're using it. Here's Giuliano, she's our head roaster and provost roasting fellow. We're in the middle of architecture. A lot of exciting things are happening. But so this is the building, this is the physical infrastructure. And what I'd like to really emphasize is that it's not just a building, the Coffee Center, it's really the human infrastructure. And so what I'd really like to acknowledge is that everything I'm going to present today has been a huge team effort. And very first matter of business is to really acknowledge my co-PI, Professor Jean-Xavier Guinard, who's a professor of consumer and sensory science, basically a global expert in that topic. And I've had a tremendous amount of fun learning from him over the past several years as we design these projects and learn how to really investigate coffee fundamentally. This one project has supported several different students. So here's Dr. Scott Frost, he was our first postdoc on this project. Dr. Mackenzie Batali, who got her PhD in food science, focusing on coffee, and is now a postdoc with us. And Andrew Cotter, who got his master's degree and graduated last year. So these are the main students who worked on this. And as Phil and Peter alluded to, we've had a whole ecosystem of other students. And here's just a few of them. I actually ran out of room to put snapshots of photos. A few of them helped principally with the work that I'll be showing here today. And I'd like to really emphasize that none of this would have happened without Breville's support. They provided the underwriting to the Coffee Science Foundation to make this happen. And we had a few little in-kind contributions from Curtis, Royal Coffee, and Java City in terms of coffee donations. But Breville's in a big font here because without their forward-looking vision, this research wouldn't happen. And so thank you for that. Okay, so what am I going to talk about today? And so this is a kind of a crazy overview because I'm trying to summarize the outputs of 3.5 years worth of research. And we've had a lot of really fascinating results. And so let me walk you through this. What we're going to do is very briefly go through kind of a little bit of brief history and motivation about this thing, the coffee brewing control chart, which I have to admit, the first time I saw it, I was very confused as well. And then part two, I'll illustrate kind of the way that scientists approach a question, which Phil already mentioned. What's the difference when you brew in a flat bottom basket versus a conical basket when you keep everything else the same? And so we have really cool data about that, and I will share that with you. Part three is another kind of warm-up project that we did. So these first two projects were kind of like initial, early, kind of quote easier exploratory projects. And the fractionation one, we're actually looking what comes out of a drip brewer versus time. And so the fancy word for that is fractionation. And we're very interested in how does that taste versus time. Okay, and so we'll talk about that. And then the bottom half, these parts 4, 5, and 6, really the main event. What we've done to really systematically overhaul the coffee brewing control chart. And on part 4, I'll share some really exciting results where we looked at the effect of roast level and brew temperature on various sensory attributes over the entirety of the coffee brewing control chart. Part 5 switches from what does it taste like to what do people like, and that's consumer preferences. And then everything will culminate in an exploration of like what will the new coffee brewing control chart look like. So that's the outline. It's very ambitious. I'll probably have to talk really quickly, but hopefully we'll get through all this in 1 hour. Okay, so first, history and motivation. And so very briefly, Peter already mentioned this. A lot of the research for the current coffee brewing control chart was based on work done in the 1950s and 60s. So here's a snapshot of Professor Ernest Earl Lockhart around 1960 or so. And he was the director of something called the Coffee Brewing Institute. And in the world of coffee science, he wrote a really seminal paper in 1957 called The Soluble Solids in Beverage Coffee as an Index to Cup Quality. And that's a fancy way of saying he was looking for a single number that would tell you how good is your coffee. Okay? And so he did a bunch of research, and then that work was compiled by Ted Lingle into the coffee brewing, or the Brewing Handbook, which I understand from the Specialty Coffee Association is still their #1 selling handbook of all time. And so what's in that book? Well, it really hinges on these 2 key metrics for coffee brewing. And since I'm going to be talking about all these, I suspect many people in the crowd already know about this, but to make sure everybody's on the same page, here are these 2 metrics. The first one is the percent extraction, or PE, and it's also known as the extraction yield. Okay, and so that's literally how much of the soluble material did you rip out of the solid coffee grounds into the liquid phase. Okay, and so coffee grounds are at most about 30% soluble in water, and roughly speaking, usually people are aiming for about 20%. So that's one metric. And the second metric is the strength. Okay, also known as the total dissolved solids, and that's actually what's in your cup. What are you drinking? How strong is it? And an average cup of coffee is 98 to 99% water and about 1 to 2% dissolved solids. That's what makes it black. That small percentage of stuff. Okay. So Ernest Lockhart's great insight was to correlate those 2 metrics, okay, the strength and the extraction yield, with what it tasted like. Okay, and so here's one formulation of the classic coffee brewing control chart. On the vertical axis is the extraction yield, it's the extracted solids, the total dissolved solids, how strong it is on the vertical axis. And on the horizontal axis is the extraction yield, okay, the percent extraction. The diagonal lines are the brew ratio, okay, how much water did you use per mass of coffee grounds. And the most important thing is superimposed on this chart are various sensory attributes. And so on the right-hand side, you see bitter. On the left-hand side, you see underdeveloped, which is often interpreted as kind of grassy or vegetal or sour. Okay. In the vertical direction, you have strong and weak. And the most important thing from the kind of modern coffee industry's point of view is that you have this ideal range right here in the middle. Okay. And that's also known as the golden cup standard. A lot of modern training teaches people you got to aim for your coffee should be in the middle of that box. That's your target. Okay. And so this was developed by Lockhart and his colleagues back in the 50s. And there's, from a modern sensory point of view, a lot of challenging aspects about it. And so the very first one is that the way it's set up is that you have these really sharp dividing lines that really falsely imply that a minor difference in strength or a minor difference in yield is going to get you in a good region or a bad region. Okay, and in reality, everything's a continuum. That should be smooth. It shouldn't have a sharp line like that. The second and bigger problem is that the chart really conflates two things that are different. In other words, it conflates sensory descriptive attributes. You know, what does it taste like, with hedonic sensory judgments. What do people like? Okay, some people like bitter, some people like sour. Okay, and so this, from a modern sensory descriptive point of view, this chart mixes those up in a way that's problematic. And then, again, this was developed in the 1950s and based actually on quantitative data that, as far as we can tell, is no longer available. So, you know, who decided that, what range is there? Like, it's we've not been able to find it over the past few years of digging. But presumably it was based on some type of consumer data back in the 1950s. And I have a couple of pictures of, you know, what it kind of looked like back then, right? The key point is it's not representative today. The coffee industry has moved on in the past 70 years. Okay. So the final big thing is that if you look at the number of sensory attributes on this chart, there's only like two words, bitter or underdeveloped. But if you've been to any modern specialty coffee event, you've probably seen this. Here's the Specialty Coffee Association Coffee Tasters Flavor Wheel. It's got about 104 different attributes on it. All these things that the specialty coffee community loves, you know, different berry flavors, chocolate, nutty stuff. And the question is, where is that stuff? Okay, how do you optimize your brewing to get certain attributes out of this? And, you know, there's no insight from the classic coffee brewer control chart. And so these are not, you know, these are not original insights to me. I think there are a lot of thought leaders in the coffee industry, including Peter, you know, had a sense that there's a lot, you know, missing here. And so back in April of 2017, I like that phrase, the dream team came together. And so because of the Coffee Science Foundation and Breville support, we were able to like start launching into this and try to answer some of these questions. Okay, and so one of the very first things we did was check out this question of flat versus cone. Okay, and so you know, Breville was supporting it, and like all these conversations with Breville, and they'd already developed this system. And I remember one of the first questions was, why do you have both? And they said, well, we think it changes the flavor. And so that's a testable hypothesis. Does it change the flavor? You know, it doesn't matter. Does it matter if you brew into a flat bottom or semi-conical? And so we thought, why don't we test this rigorously? Okay. And so the questions we want to ask specifically is, how does this basket geometry affect the extraction? How does it affect the flavor profile? And so the way we did this was, you know, it's kind of a simple idea. We held everything constant, everything, same coffee ground, same ground size and everything, same water delivery, okay? And we just changed the basket shape, okay? So all these things, the water composition, feed temperature, all the brew ratio, keep those the same, and just swap out using the Breville precision thing here, just the conical basket or the flat basket. And we also changed the grind size. So we had a coarse grind and a fine grind. And so these are kind of our independent variables. And then our dependent variables, we just want to see what happens. And so we measured the strength, the TDS, we measured the percent extraction, mass flow rate, and very importantly, the sensory profile. What did it taste like on the way out? And so I mentioned we changed the grind size, and so we used some pretty different grind sizes. So this is on a Malcanig, Guatemala, and we weren't doing very small changes in grind size. We actually did 25% and 50% change in the grind size for a couple different roast levels, okay, of a kind of a generic Central American washed coffee. And so here's some of the earliest data that we took. And so this is a measure of the TDS, the total dissolved solids, so how strong it is, as a function of time. So imagine just holding a test tube under where it's coming out and grabbing the first few seconds and then grabbing with a different test tube the next few seconds, and then measuring the total dissolved solids. And right away, you can see at a glance that the basket geometry has a huge impact on the dynamics of the extraction. So the red points here are the flat bottom basket. And you can see that the first few seconds of stuff coming out of flat bottom, really high TDS, very strong. Okay, and then it tapers down. In contrast to the conical one, it actually started low and then went up and then decreased. Okay, so just, again, everything's the same, just geometry of the basket and the dynamics of the extraction are changing dramatically. And we want to be very thorough and make sure we're nailing down potential confounding factors. And so here are some histograms showing the temperature profile here. And the bottom line here is that like the conical stuff came out a little bit warmer than the flat bottom stuff, presumably because of the different surface area volume ratio of the different geometries. Okay, but it wasn't a big difference. Okay, and so here is a box plot of a whole bunch of different brews. And so what we're looking at here is kind of a snapshot where we did a brew, and then at the end of the brew, what is the strength? And so here's the total dissolved solids on the vertical axis, and here's a medium grind size, and here's a fine grind size, and this is for the flat bottom, and here's medium and fine for the semi-conical. And so you can see at a glance that actually, right, if you want to change the total dissolved solids, it's more effective to change the basket geometry than to change the grind size, at least over this range that we changed. Okay, and so in other words, a 25% increase in grind size had a smaller impact than just swapping the geometry. Okay, you can also see that there's a bit of variability. Any drip brewer under the sun is going to have a little bit of channeling. So sometimes the water always follows the path of least resistance. So sometimes you have more channeling, sometimes you have less. So there's a bit of scatter, but this is like representative data. Okay, so clearly the brewing geometry affects the total strength of the coffee. Big question is, does this matter for flavor? Okay, and so there's a bunch of ways you can do that. And what I'd like to walk you through here is one style of sensory testing, which is called discrimination testing. And if you've ever done a triangle test, then you're familiar with this. And here's a basic idea. You're presented with 3 cups of coffee. 2 of them are identical and 1 is different. And you taste them blind, and the question is, can you tell which one is the odd one out? Okay. And so if they're wildly different, 2 have cream and 1 does not, then that's easy. Okay. But for things that may have a small difference in TDS based on the basket geometry, this is the type of test you want to do to see if you have statistically significant chance of telling that there's a difference. Okay. And so in this type of test, it's binary. You either get it right or you get it wrong. So it's a yes, no. And you have here's the complicated thing is that you have a 33% probability of getting it right just by chance. Okay. And so if you have one person do it and they get it right, that doesn't really tell you anything. They might have got it right just by chance. Okay. And so the way to think about this just very quickly is to go through something known as a binomial distribution. Okay. The classic example is flipping coins. If you take undergraduate statistics, you'll get to study this in detail. A simple question is if you flip 4 coins, what are the odds that you'll get 2 heads and 2 tails? And if you guessed 50%, that's wrong actually, all right? It's actually less than that, because if you look at the possible outcomes, and here's kind of that chart right here, only 6 out of 16 possible combinations give you 2 heads and 2 tails, and that works out to 38%. Okay? And so this is for flipping coins, right? If you do, say, 6 triangles, like for example, if you're trying to get Q certified, okay, what are the chances that you'll get 6 of them right, just randomly, okay, just by choosing randomly? You can see that here the most probable outcome is you get, if it's 1 out of 3 for a triangle, then 1/3 will have 2 out of 6 correct, okay? But if you get 5 out of 6 correct, which if I believe that's the minimum for getting Q certified, there's actually a 1.6% chance of getting that right just by chance. So to do discrimination testing very rigorously, you have to use a very large sample size. And so here's what if we do n equals 45. So let's imagine we had 45 consumers come in and test different triangles. If it's no better than random, then what you'd expect is that you get 1/3, so 15 out of 45 would get it right. And here's the probability distribution that corresponds to that. Okay. And so basically, let's say you bring in 45 consumers, you have them taste coffee, and 16 of them get it right, and the rest get it wrong. That's no better than random. It's right in the middle of this probability distribution. Those products are not distinguishable. If, let's say, 40 out of 45 get it right, okay, that is not at all going to happen by random. That means that you had a statistically significant difference. Those products are distinguishable. Okay, so we did this with the flat bottom versus conical, and we used sensory isolation booths. This is one of the nice things in the Robert Mondavi Institute. And so it's a little booth. It's got red lights, so you can't even really see the coffee. It minimizes expectation bias. You're isolated, so there's no context bias. You're not tasting next to your buddy. Okay. The 3 coffees slide in, and we had our consumers tell us which one's different. Okay. And so we had a 2x2 factorial design, which is a fancy way of saying we had actually 6 different comparisons. So we can compare a flat bottom basket versus a conical basket, both with medium grind size, for example, or we can compare a cone with medium versus a cone with fine. We can do the cross products. And so we recruited 45 totally untrained consumers to serve as panelists. Okay, and they each got presented with 6 experimental triangles, plus a control triangle of what we thought was an extremely different coffee of a dark versus a light roast. And just for this kind of quote simple test, we actually ended up serving 945 distinct cups of coffee. And so here are the results. And so again, here is our probability distribution for a probability of 1/3 chance getting it right for 45 people. And our obvious difference, our light roast versus dark roast, is way out here. Okay, we had 39 out of 45 successes, and so that's what we expected. Okay, it means 6 people even got that wrong. Okay. Our fine versus medium in the flat. Okay, so this is 2 different grind levels in the same basket. 18 out of 45 got it right. No better than random. Okay, and so that grind size difference at least had no statistically significant impact. Flat versus cone. Okay, same grind size was 25 out of 45, so way over here. Okay, at the very tip of this, and so that's actually at the above the threshold for statistical significance of usually use alpha equals 0.05. Okay, so is there less than a 5% chance it would happen by random? And the answer is yes. Okay, and so we can look at all the different comparisons, and the bottom line, I know there's a complicated figure here, but the bottom line is that when we just compared grind size, it was not statistically significant. Okay, when we compared basket geometry, it was. And so that was, a lot of work just to see, is there a difference? Can people detect a difference? And the short answer is yes. It matters if you use flat versus conical, because it changes the strength, and it changes the strength so dramatically that even totally untrained consumers can tell the difference. Okay. We did a lot more work, okay, on sensory descriptive analysis of consumer preference testing. And I'm not even going to walk you through this because I want to get to the more interesting things here. But we did a lot of consumer preference testing. The bottom line of this stuff is that both an expert panel and consumers, in terms of their preferences, found differences, statistically significant differences, again, just based on that basket geometry. Okay, and so, and based on the basket geometry, you can go and maximize how sour and how citrusy it is versus how bitter it is. Again, same coffee, just change the basket. Okay. And so these details, and by the way, this we're very proud of this one. This is Scott Frost's work, got published on the cover of the Journal of Food Science. There's an SCA magazine article about it, and there's this Journal of Food Science article that you can read about it in more detail. Okay, so that's one project. All right. Second project, second warm-up project, was thinking about fractionation. And by the way, I'm going to stick around for any questions that you have at the end of this. I'll be happy to answer any questions about anything. So feel free to take notes and ask me lots of questions at that. So part 3, fractionation. Okay, and so let's go back to this question here again. Right. So here is, we changed the basket geometry, and the profile, the TDS, the strength is dramatically different. And so a very natural question for us was, well, gosh, what flavors are coming out when? Okay, so we know that the coffee has bitterness, it has sourness, it has all these different sensory attributes. Do they come out at different times? Like when is it just simply correlated with TDS? How does it begin? So we wanted to answer that question, right? And so and what we did, this is now work of led by Kenzie Batali, okay? And we set up a sensory study where we fractionated the coffee. And we actually used a bigger commercial brewer, this is a Curtis brewer, and it's a pretty simple question, okay, or pretty simple conceptually. It's a lot of work logistically. But basically just imagine holding a beaker under the spout or under the brew head for the first 30 seconds, and then the next 30 seconds, and then the next 30 seconds. And so you keep swapping them out, and you just catch it. And so you can see here just visually that the first 30 seconds, at least in this brewer, are very dark, very strong, and then it gets progressively weaker as it goes along versus time. And so that's once we collected all those individually, then we can do physical measurements. We can measure the mass, the total dissolved solids, pH, etc. And then we can also do sensory descriptive work. Okay, so first focusing on the physical stuff, for this brewer in that basket shape, here's the total dissolved solids versus the fraction number. Okay, and you can see that again, because of channeling, even in this commercial brewer, we get different, there's a little bit of scatter, but on average, it started off really strong, up here around 3% TDS, and then decreased as it went down in time. Okay. The coffee brew mass dispensed, okay, so even though this first fraction is really strong, there's also not a lot of it, okay. Initially, the water is going in and absorbing into the coffee ground, so not a lot is going through. The stuff that does drip through is very strong. But you can see the mass delivered actually increases, and then it tapers off as the last few drips come through. Okay, and so now to answer the question, what does it taste like? We turn, not discrimination testing, but we turn to descriptive analysis, okay? And that's really to identify what flavor profile is present in the food product, okay? And so here's one representation of the coffee taster's flavor wheel. Molly Spencer worked with Jean-Xavier Guinard a few years ago and actually did some statistics that underlaid that. And the whole idea is that you get a panel of trained experts together that all agree on what, you know, if you use the term rubber, what does rubber mean in this product, okay? And so they actually spend a tremendous amount of time familiarizing the members of the expert panel, okay, with different aroma and taste reference standards. And so here's a few different pictures. Here's a coffee cup with molasses in it. Here's one of some rubber bands to indicate rubber. And all the part of training panels to agree kind of numerically, what does a score of 50 mean or a score of 25 mean when we're talking about rubber or sour or bitter. Okay. And so for the various attributes, here's a whole bunch of things that you could look at, you know, sweet, sour, bitter, salty, umami, astringent mouthfeel, and then all these different flavors that people, especially coffee world, especially care about: honey, fruity, nutty, okay, cocoa, things you probably don't want, rubber, okay, meaty, brothy, things like that. Okay. And so they calibrated the panelists. There's a snapshot. This is up in Robert Manavi Institute. And then the way it works is, again, using these sensory booths. They have one sample at a time. They're presented with it. They taste it, and then they have an unmarked scale where they go and say, oh, it's this sour or this much sweetness, okay? And then you end up collecting a lot of data, okay? So I'm zooming in just onto the results, okay? So there's a lot of like internal calibration data, but here's the question: how did those fractions, what do they taste like versus time, okay? And so here's a plot of the attribute intensity, okay, averaged over all of the different panelists. And by the way, they're tasting the different fractions in random order and blind. So they're not tasting the first fraction, then the second fraction, the third fraction, tasting in random order every single time. Okay? They have no idea what they're tasting. And so this is averaged over the entire panel. Here's the bitterness intensity, okay? And this kind of behaves the way you'd expect. The very first fraction was extremely bitter, intensely bitter. And then that bitterness decreased as you go to the weaker and weaker TDS samples. And many attributes acted exactly like that. So here's sourness, okay? Here's smoky, burnt, it also decreased. Astringent decreased. Alcoholic, winey, decreased, okay? And so that's kind of what we expected, you know, and the stronger the total dissolved solids, the more stuff there is, so the more intense the sensory attributes should be. But here's what really shocked us, okay? Some of the attributes actually increased with time, and they're important attributes. And so the first one that we point out here is sweetness, okay? And so again, you know, we're not adding sugar or anything. This is black coffee, but there is perceptible sweetness, and it was very low for the very early fraction, and it increased with later fractions that got weaker. And here's another one, tea/floral, also increased. And so just to really emphasize that, so here's a bunch of graphs. Here is bitter intensity up here in the left corner versus total dissolved solids. These are the different fraction numbers, and it behaves, like I said, the way you'd expect it. The higher the TDS, the more bitter it was. But here's one that a lot of specialty coffee people care about. Here's sweetness. It behaves exactly the opposite. Here's sweet intensity versus total dissolved solids. The negative correlation, it decreased. And so you can look for the correlation coefficient, which is kind of the slope of this line. And that's plotted here. Here's the correlation coefficient versus the different attributes. And many of the attributes decrease as TDS decreases, so it behaves like this, okay? So bitterness, rubber, smoky, all these things in here, all right? The stronger it is, the higher the attribute, okay? But then the ones that a lot of people care about, sweet, tea, floral, cereal, honey, fruity, those actually were negatively correlating, all right? So they got stronger at the end of the fractions, okay? And so just to recap, if you want to maximize the sweet taste, the fruity taste, the honey taste, the tea floral, the cereal flavor, you probably should throw away the first few fractions and only serve this down here, okay? In contrast, if you really want to have high and everything else, bitter, salty, all these other things, then you want to have the first few fractions and throw away these guys. Okay, and so this is very counterintuitive. It's very, I should really emphasize this, very unusual to have a concentration go down in a food product, but the sensory attribute increase in intensity. Very, very unusual. And so what we had, what we wanted to test was a possible hypothesis. Perhaps the concentration of natural sugars is higher in later fractions. Okay. And so Kenzie Batali, who again, brilliant student who led all this research, she went and worked with Carlito Labria in our chemistry department, who's a world expert in mass spec, and did some pretty sensitive monosaccharide analysis. And so monosaccharide, that's a fancy word for sugars. Okay. And so without going into a tremendous amount of detail, she went and analyzed lots of different monosaccharides that are known to, impart sweetness through taste. Right. So here's almost a scary looking chemical formulas for all the different sugars, different types of sugars that were looked at. Okay. And this is a tremendous amount of work. Right. And so it's not like Star Trek, waving a tricorder over a sample and it spits out a number. Right. So to do this is a lot of labor on a pretty expensive piece of equipment. There's a lot of fancy words here, a lot of chemical pre-treatment steps to get to the final thing that we need in the mass spec. And so just zooming right in, here's the final analysis that we got, okay? And so here we're looking at the concentration in milligrams per milliliter of a whole bunch of different sugars, monosaccharides, versus fraction number. And so the first thing you can see at a glance, all right, so the different colors here represent different sugars. The first thing you can see at a glance is that that hypothesis is wrong. It's not that there's somehow the sugar concentrations or monosaccharide concentration is increasing the later fractions. If anything, it's going down, okay? So no big, increase. So that hypothesis is not verified at all by the chemical analysis. A real important thing to note is that these concentrations are in milligrams per milliliter, right? So even though they're present, it turns out they're present there at extremely trace amounts. So here, this red line around 3.5, that's the threshold for human sensory perception of sucrose and fructose, which are, two of the most famous sugars. All the concentrations are way below that. So in other words, you can't really explain it, or you can't explain the perceived sweetness in the black coffee as a function of the presence of monosaccharides. And so that's really at odds with a lot of the kind of conventional wisdom out there. You can find a lot of things on the internet talking about how there's like sugars left over from the roasting process that contribute to sweetness. At least in this sample, that is not at all consistent with what we're seeing here. And so that really raises a question, or I guess the conclusion is that perceived sweetness has nothing to do with the oligosaccharides here. The question, but we do, our expert panelists do perceive sweetness. So what is that from? Well, there's a couple of different hypotheses, okay? It could be that there's, they're all equally sweet, but at the lower fractions or the earlier fractions, there's so much bitterness and there's so much sourness, it really just overwhelms the sweet perception. It doesn't break through the sweetness, okay? Another hypothesis is that there are other flavor molecules that your brain associates with sweetness, okay, that are there at higher concentrations later on. And so, for example, if you smell apple aroma, okay, and then chew on a potato and you're blindfolded, it'll taste like an apple because of the aroma, right? That's a way that your brain is tricked into perceiving different things. And that potato will taste sweeter just because of the apple association. So that's discussed in more detail. And so here's Kenzie's paper. It came out in the Journal of the Science of Food and Agriculture. And there's also an SCA magazine article, Less Strong, More Sweet. Okay, so now on to the main event, okay? And so I'm really proud of both those papers, the 5 versus code and the fractionation. But again, we were motivated at the beginning to understand how do we update the coffee brewing control chart, okay? And these first 2 papers provided us with a lot of experience on brewing and with how it behaves, and a little bit of insight that there's some odd things going on here that actually, for example, you get more sweetness at lower TDSs, okay? And so here's the main research objectives. We want to update the brewing control chart, okay? And the main hypothesis, just to be very explicit in a scientific way, is that coffee brewed at different positions on the chart, okay, will have different sensory attributes, and the consumers will have different preferences for coffees brewed at those different positions. So here's the main experimental design, okay? And this is now going to be an order of magnitude more work than the previous 2 experiments. Here is, again, the same format of the chart. Here's the strength versus extraction yield. And we're going to do a 3x3x3 factorial design. And so the basic idea is we're going to brew coffees at kind of each of the 9 main quadrants of the coffee brewing control chart. So that means we have to do 3 levels of TDS, and we chose 1, 1.25, and 1.5. 3 levels of extraction yield, 16%, 20%, and 24%. And for this first experiment, which was led by Dr. Scott Frost, we're going to do the same green coffee, but at 3 different roast levels, light, medium, and dark, to see how universal it is for 3 pretty wildly different roast levels. Okay, and so just to really emphasize, that means we have 27 different experimental brews that have to be tested very rigorously from a sensory descriptive perspective. Okay, and so we have to precisely control the TDS and the percent extraction to make that happen. And so very briefly, the way we do that is we use something that we call the water pulsing duty cycle. And so again, we're serving panelists, and so we have to use a commercial brewer to be able to deliver enough coffee for testing. And so this water pulsing duty cycle, what we can do is deliver the same amount of water, okay? So our brew ratio is the same, but take advantage of having the water on, off for a while, on, off for a while, so that the coffee has more time to extract in the brew basket, okay? And so you can see here, like we can do 1 pulse of water for 160 seconds, or we can do 2 pulses over a total brew time of 320 seconds, but the water is on for 80 seconds each. So again, the same amount of water delivered, but different total amount of brew times. And so here is a cool chart showing the different, water pulsing duty cycles that we use to be able to cover the entirety of the coffee brewing control chart. And this is work that actually Jessie Liang, one of my PhD students, and Xian Lim helped work on to develop this. And so we actually have another paper in preparation just focusing on this. But here I'm going to zoom in on the coffee brewing control chart and the results. Okay, and so here are experimental data showing the TDS and extraction yield for all the brews we did for the 3 different roast levels. So each of the different colors here represents one of our target goals. Remember, we're trying to hit TDS and percent extraction in each of those quadrants. And so over here, you can see that we actually did, I think, or the students did, Scott did an excellent job of getting reproducibility for our target. There's always going to be a little bit of scatter, but most of the time we hit it pretty right on, including, which I guess a lot of specialty coffee people tell me that the ones down in the bottom right corner are the hardest to get. And the trick basically is you have to use a longer total brew time, kind of silly long brew times, 10, 15 minutes, and you can get down in that corner. Okay, and so we brewed the coffee, right? And again, we want to do the same thing, descriptive analysis with a trained expert panel, right? And so, and here's a snapshot of all the different sensory references that we used. Here's a lot of the flavors that we care about, blueberry, brown spice, citrus, chocolate, all the tastes that we care about, sweetness, sourness, bitterness. And so all the details of what we use for the sensory references are available in the paper. And so we trained the expert panel. We brewed all 243 brews, all over the coffee brewing control chart for all 3 different roast levels, and we get an inordinate amount of data. And so here's a very small snapshot of what some raw data looks like. And so here's different individuals of these different numbers. Here's different coffees, okay? And so here's a light roast, for example, at a 1% TDS and a 16% extraction. And here are the attribute intensities as judged by that one panelist for that one coffee, for that one sample. And we test all the brews in triplicate, so we just get tens of thousands of data points. I forget what it was for this study. It's probably 30,000 data points for this one particular study. So what that means is we have a tremendous job in reducing all those data into ways that from which we can extract meaningful trends. And there's lots of ways we can do that. And so here's a couple of the more complicated ways. Here on the left, this is showing attribute intensity. Okay, so the higher numbers are bigger for various attributes. Okay, and then the different columns here represent the different either roast levels, so light, medium, or dark. The different total dissolved solids, so 1%, 1.25, or 1.5, and this column is percent extraction, 16, 20, or 24. So this is looking at individual parameters here and seeing. Okay, you can think of it as like if we hold everything else constant, is there a statistically significant difference in these letters, all these A, B's, and C's and whatnot? If those letters are different, then there is a statistically significant difference. Okay, and so at a glance, you can see that roast changed everything. Okay, or almost everything, and that's not surprising. And so, it's not a surprise to anybody that if you change the roast level dramatically, you can change the flavor profile. What's more interesting is that at a constant roast level, right, if you change the TDS, okay, you change bitterness, you change astringency. Some of these flavors, like blueberry, for example, there was no statistically significant difference in blueberry, presumably because this particular coffee we used, which again was kind of a Latin American, traditional washed coffee, didn't have much blueberry flavor in the first place. And percent extraction also changed it. So this is one representation looking at individual attributes versus individual parameters and looking for significance. Over here on the right is a more complicated way. This is something called principal components analysis. Okay, and so you can see that we have the different colors here represent different roast levels, and the numbers represent different extraction yields and TDSs. And with a principal components plot, you're looking for separation between the things, okay, or between the different items. And you see the light roasts tend to be up here, mediums over here, darks over here. And if you look carefully at this, you'll see that there's also separation based on the middle number, which is the brew strength. Okay? And then over here is the corresponding attributes chart. And you can see that, one way to look at this is like, if I want to maximize sourness and citrus, these products up here have the most sourness and citrus, for example. The most earthy and roasted ones are down here. Okay? And so this is kind of a complicated way of looking at it. And again, I'll emphasize that the details of this are present in the peer-reviewed publications. Let me zoom in on, I think, what's the most intuitive way to understand or make sense of all the complicated data, which is something called response surface methodology, which is a fancy term, and a better way to think about it is we're going to make some contour maps. Okay, and so here I'm showing pictures of, this is a map of Pittsburgh, and this is San Francisco, and so they're maps, and the color represents the elevation, okay, the altitude. If you've ever been to San Francisco, you know there's some mountains in the middle. If you've ever been to Pittsburgh, you know that downtown is actually at the lowest elevation, okay, right next to the rivers. And so we want to do the same thing for these sensory attributes, okay, versus the coffee brewing control chart. And so we had all the data, and we were able to do that. And so here's a few of those, right? And so here is the particular attribute of sourness, right? And so here's the total dissolved solids versus extraction. And so we take our noisy data, and we basically fit a contour map through that. And so in these graphs here, red is high intensity, green is low intensity. And so here you can see, and this might be intuitive for a lot of people, that if you want to maximize sourness, you want to, for this light roast, for example, you want to have very high TDS, so very strong, and also a low extraction yield. And so it's maximum up here, and perceived sourness was lower down in the opposite corner. And you can see that trend was actually conserved across roast levels. So medium roast and even dark roast had that same feature where sourness is maximized in the top left corner of the chart. Bitterness behaved in the opposite direction. Here, bitterness is actually maximized in the top right corner. So that means you have to extract as much as possible and go to a very high TDS, okay? And again, it's conserved across roast levels. That's where you got the maximum bitterness, okay? And both those are kind of what I think a lot of people would expect, higher TDS, you definitely expect that. And I think a lot of people associate under extraction with sourness, so you kind of expect that. But then going back to our fractionation results, of course, we're very intensely interested in sweetness. And so sweetness is upside down. Okay, sweetness is upside down. And so what we see here is that sweetness, perceptible sweetness, is actually maximized in the bottom left corner. So it's actually at lower TDS and lower extraction yield. And now, again, we see that same trend independent or regardless of roast level. And so that was actually, we were really excited when we saw that. It was consistent with our fractionation results. These samples we're looking at right here are not fractionated. This is just the total brew, okay? So even in the finished final brew, okay, at low TDS, at low extraction yield, that's how you maximize the sweetness. And so those were, and we have, I should emphasize, we have sensory attributes for many different attributes, all those different things we measured. Here, this is a graph that summarizes it over all roast levels. So in some sense, let's forget about the roast level difference. Let's average everything together, okay? And it turned out there were 13 sensory attributes that were statistically significantly different versus TDS and P, irregardless of roast level. And so you can see those here. We've got sourness, citrus behaves the same. Astringency was increased at highness. I already showed you bitterness, high over here. And then there's a whole bunch of other flavors that their magnitude of change of the sensory attribute was not as big, but still statistically significant. And then sweetness is the only one average over roast level that was maximized down in that bottom left corner. Okay, and so that was a tremendous amount of work, hundreds of brews, 27 samples, months of work to come up with this. And I remember we met with Breville somewhere in the middle of this project. And they said something to the effect like, "This is excellent, but what about brew temperature?" And so, oh my gosh, okay, let's dive into brew temperature. So basically, we did the whole experiment again, right? And I'm really glad that Breville insisted on that because, man, we got some really exciting results. And so the idea is let's do the whole thing over again, but instead of changing roast level, let's just use one roast level. But now let's vary the brew temperature, but keep the TDS and extraction yield the same, okay? And so same idea, 3 by 3 by 3 factorial, all right? And the 3 brew temperatures we're gonna look at are 87, 90, and 93. And so some of you might be familiar with the Gold Cup standard or Gold Cup brewers. I think, I believe they have to hit 92 degrees Celsius within the first minute of the brew. There's this cutoff, right? And so we're very interested in like, well, does that temperature cutoff really matter? I mean, is it okay if you brew at 90 degrees instead of 93 or about 87, right? And if you go online, you can find lots of references to different people saying like, oh, you got to brew at a certain temperature or it doesn't taste right. So we're going to test that quantitatively. And so how do we do this? I really want to emphasize that we changed the brew temperature, okay, as an independent variable, but we changed other things like the grind size or the duty cycle, water pulsing duty cycle, to get to the same target TDS at a given brew temperature. So here, this is really illustrated here. Here's at 93 degrees, so our incoming water is 93 degrees Celsius, and we're trying to hit this target zone right here. And so we used a different grind size and a different total brew time to hit that. And then over here, we used 87 degrees Celsius, okay? And we just changed those other parameters, got the same on average TDS and extraction yield. So I just really want to emphasize, we changed the brew temperature, but changed other things, so we got to the same destination. And the underlying hypothesis here is that maybe the different temperatures extract different, flavor molecules differently, and that's what matters. And so that's what we wanted to test. And so here's a similar one of those plots, so showing that we have pretty good reproducibility over and over again with TDS and extraction yield. And again, here's similar results. So here are here's total dissolved solids, here's percent extraction, here's temperature, and here are the different attributes: bitter, sour, etc. Okay. And again, if you have letters that are different, that means there is a statistically significant difference. And so, at a glance here, you can see, wow, the total dissolved solids matters a lot. A lot of these attributes have a big difference or a statistically significant difference based on the total dissolved solids. Okay. And again, if there's blank, that means there's no statistically significant difference. Percent extraction also mattered, not as much. Okay. But several attributes did. Sourness mattered, black tea mattered, berry mattered. Okay. Look at temperature, right? It's almost empty. It's almost empty. So when you held TDS and extraction yield constant, only one popped out. Okay, nutty, and we think that's actually something called a type 2 error. Basically, it just happened by chance. Right? Whenever you have a cutoff of 5% probability of happening randomly, sometimes when you test as many attributes, you get one of those. So at a glance, you can see, wow, it looks like actually temperature didn't matter much. And again, here are some principal component plots for those of you who are familiar with looking at these. And this is really complicated here. The first number is the temperature, next one is the TDS, the next one is extraction yield. And if you stare at this graph long enough, what you'll see is that, yes, there are trends with the TDS, big trends with the TDS, big trends with the percent extraction, and the temperatures are scattered around randomly. There's no big trend with temperature. Okay. And probably, so that's a complicated way of looking at it. And here's my favorite way of looking at it. So these are some spiderweb plots that Kenzie put together. And so many of you, I think, have seen these before. And so on the left, what you're seeing is that the further the radial distance is from the center of the spiderweb, the bigger that intensity was. And so, and the different colors here correspond to different TDS values. So blue is 1%, red is 1.25, gray is 1.5. And so, and this one is a constant TDS, constant temperature, but different percent extractions. And here's different temperatures. And so here you can see, again, TDS matters a lot. There are many different attributes that change in intensity by changing the TDS. Not as dramatic with percent extraction. Several attributes changed, but not as many. And then again, look at this. It's almost identical. You can, like, the plots are almost superimposed. So changing the temperature, at least over this range, at constant TDS and constant percent extraction, okay? the temperature didn't matter, right? A trained panel, expert tasters could not tell the difference at constant TDS and constant PE based on what brew temperature you used, okay? Provided you got to that same target TDS and PE. And so that was not what we expected. I mean, we did all that because we thought brew temperature would matter a lot, right? It turns out it didn't. So this same way of, summarizing total dissolved solids and PE in the coffee brew control chart is valid. And again, for this sample, again, we did the whole experiment again, so we got very similar results from the roast experiment, where we have attributes that, for example, sourness maximized up in the top left corner, bitterness up in the top right. Okay. And so the roast stuff was led by Scott Frost, and that was published last year in the Journal of Food Science. Okay, so we did excellent work on that. And then Kenzie Vitale, who was our PhD student, she graduated with her PhD at the end of 2020, is now doing a postdoc with us. So she published this last year in Scientific Reports. And I'm really proud of this. It was actually, I checked this morning, it's been downloaded 29,000 times, and it was in the Scientific Reports top 100 for the year 2020. And so that's a huge coup for Kenzie for this, this hard work that she did. And so I think she was like number 48 or 49. Number 1 last year was apparently a paper about sex chromosomes for turtles, which shows that, I don't know, I guess people have odd tastes. But it's really impressive that I think that a paper about coffee and the fact that brew temperature, doesn't have this result, or doesn't have this effect that people kind of, commonly talk about, really struck a nerve. Okay, and so it's really cool to have impactful work like that. Okay, so I have about 15 more minutes of stuff, so hopefully we're doing okay for time. Everything I've talked to you about the coffee brew control so far has been, what does it taste like? Okay, but many of us who care about selling coffee to consumers, the question you really should be asking is like, well, what do people like, right? And so, you control what it tastes like, but then a different question is, what do they like, right? And so the fancy phrase for that is consumer preference testing. And so this is work led by Andrew Cotter, okay? And so he is the gentleman right there pouring some coffee in the back. And this is different. So you take untrained panelists, and we selected, we sent out like 4,000 emails and eventually recruited a couple hundred people, and we finished with about 118 people whose only selection criteria basically was that they liked drinking black coffee. Okay, so we threw out people who preferred putting a lot of sugar or cream. All right, I guess threw out people who didn't like drinking black coffee at all. All right, and so we ended up with 118 people, and we we tested everything over the coffee brewing control chart. So we ended up serving with 118 consumers. I think there's a typo, it should be 27 here, not 29. About more than 3,000 cups of coffee. And we did it over several weekends. It was a big project, okay? And so here we're using the Sensory Theater at the Robert Mondavi Institute for Wine and Food Science. And it's set up where you can have like little isolation booths. These are people who are recruited to come in and taste the coffees, and they had to come back 3 times. And so what do we ask them? We served them the same coffees that we used in Kenzie's experiment. So different TDS, different P, different brew temperature. Okay, and we asked them to judge it using a 9-point hedonic scale. So how much did they like it? And we asked them about the brew serving temperature, the flavor intensity, the mouthfeel, the acidity, and we asked them a bunch of other stuff like demographics and things like that. Okay, and so just to orient you, if you're not familiar with this, here's the 9-point hedonic scale. You might have seen something like this before. It goes from 1 to 9. 9 is like extremely, 5 is neutral, and 1 is dislike extremely. Okay, so high scores are good, low scores are bad. Just about right scales or jar scales, you ask about a particular attribute. So here, for example, please rate how you feel about the flavor intensity, and they can say, oh, it's too weak, it's slightly too weak, it's just about right, or it's too strong or much too strong. Okay, and then check all that apply. They're not trained panelists, so we just gave them this checklist. And if they got a really strong nutty impression, then they can check nutty, or if it's really sour, they can check sour. Okay. Okay, and so that's what we asked. And again, yet again, a whole another set of brews for this. And so here's our charts showing that we, on average, we're able to do pretty well. Okay, and I should really emphasize a tremendous amount of work to logistically, just control the serving temperature, brew it to the same target, serve it to all the consumers showing up. 3,000 cups of coffee. It's quite a bit of work. Okay? And so here's some results. So here are the jar scales, the just about right scales, for the effect of total dissolved solids. Okay? And so here, let's focus on flavor intensity first. And so here's low TDS, medium TDS, high TDS. Okay, and it behaves kind of the way you expect, right? So yellow is just about right. Red is too much. And you can see as you go from low TDS to high TDS, the people saying, oh, too much, it's too intense. Okay, that increases. And simultaneously, people saying it's too little intensity also decreases. Okay. Same thing for acidity, all right? So the perceived acidity increased, is too much increased with TDS. Mouthfeel increased a little bit with that. Serving temperature had a little slight trend downwards. Okay. Here's extraction yield, okay? And so in terms of a flavor intensity, okay, not as big of a difference. Okay? Where it made the biggest difference was on what, again, these untrained consumers responded to was the acidity. So you can see as you increased the extraction yield, the amount of consumers responding too much acidity decreased, went from 36% to 24%. Okay? And then mouthfeel and serving temperature, not much of a big difference. And again, here's brew temperature, okay? And this is low temperature, medium temperature, high temperature, right? 87 degrees up to 93 degrees. And you can see flavor intensity didn't change much, acidity didn't change much, mouthfeel didn't change much. The only thing that really changed was adequacy of serving temperature. We had that as an internal control. And so you can see that actually just serving it, and we did our best to try to let it cool down. We had a mandatory cool down period. We had people measuring things or like representative samples to get data on that. But you can see that as this internal control is consistent, that when we use 97 degrees, you know, more people indicated, ooh, like that initial sip was a bit too hot, right? So this is some of the jar scale data, right? And it's very consistent with what our expert panel said, that TDS mattered a lot, extraction yield mattered somewhat, and the flavor, the temperature, brew temperature, didn't change it as much, okay? And so those jar scales were averaged over the entire 118 consumers. And so for this question of like, well, you know, what brewing profile maximizes liking, this is a classic problem in consumer science, okay? The current coffee brewing control chart says there's basically one ideal. You know, there's one range of TDS and extraction yield. There's one size fits all, right? And the big problem with that is you might alienate many of your consumers because maybe they don't actually like that one size fits all. The opposite extreme is like every consumer walks into your cafe, you give them a different cup of coffee tailored to that one individual. And so clearly that's logistically impossible. And so what you end up doing in practice is consumer preference clustering. So you try to make an optimal number of different products that will keep everybody happy. They can come in and choose something that's closest to their preferences. And so to do this, Andrew did something called hierarchical agglomeration clustering. All right, and so this is a dendrogram showing all 118 individuals. And so down here at the bottom, there's 118 of these little lines. And the way to think about this is basically you take all of the data from a certain respondent and you ask which other respondent is most similar out of the other 117. And so right here, for example, this guy was most similar to this guy. And then you take all those pairs and then you find, okay, which pair was most similar to another pair? And that's going up the chain right here. So this pair was closest to this pair. And you keep going all the ways up, right? And so it's a little bit arbitrary how you break things out. But we essentially identified over our set of 118 Northern California, mostly college age testers, right? We had a range of ages, but like, you know, they're all associated with the university. We found two different preference clusters, cluster 1 and cluster 2. So 51 and 67. Right? And so how do these two clusters differentiate? Again, here's the kind of complicated way of looking at it. Here's a PCA plot and versus the different respondents to each arrow. Here's a different person. Cluster 1 is red, cluster 2 is blue. And when you look at the different product attributes, what you can see is that there's a huge variation based on TDS, okay? And so the different colors here represent the different TDS values. So the red's all 1.5, yellow is 1.25, green is 1.0. And so you can see a lot of the differentiation has to do with the TDS and the rest of it has to do with the percent extraction. Okay? And down here are some bar charts showing kind of for the different clusters, what the mean overall hedonic score. Remember, 9 was extremely like, 1 is extremely dislike. And so you can see that these two clusters they actually behave differently with respect to TDS. Cluster 1 preferred stronger, cluster 2 preferred weaker. Okay, and then cluster 1 had no statistically significant difference on extraction yield. Cluster 2 did, and then neither cluster had any statistically significant difference based on brew temperature. Okay, and so again, going back to our contour map idea, we can take the data and plot it versus total dissolved solids and extraction yield, and so and here's the results. And so these are a little complicated, so let me walk you through them. So here's first in the middle, let's focus on this. This is cluster 2, okay? And what you're looking at, again, the contour plot here is not what does it taste like, this is the hedonic score. So red is good, blue and white is bad. And so cluster 2, which I've used this perhaps misleading name, I call them sweet lovers, because where do they like it? They like the brew down here at low TDS and low extraction yield, which you remember from our sensory descriptive work. That's where Scott and Kinsey identified that the coffee tastes maximally sweet. Okay, and so I have a little footnote here because Jean-Xavier, my colleague, doesn't like these names, right? Because it could also just be maybe cluster 2 is bitter haters. Okay, and so a more accurate name would be the cluster that happens to like TDS and percent extraction yields, where sensory descriptive analysis has identified sweetness as being maximal in intensity. That's a really long name, so that's my unofficial nickname for them. And so you can see they're like, whoops, they're down here. They really strongly dislike the ones that were identified as being intensely sour, and they don't like the ones over here at very high extraction yield. Cluster 1, again, my perhaps inexact nickname for them is extremophiles. They have a more complicated contour map, and it's kind of a saddle-shaped node. And so it's actually maximum over here and maximum over here. So it's kind of shaped like a horse saddle. And they actually dislike these coffees down here. They didn't like the sweet coffees. They like the ones that were the too extreme in extraction yield and higher TDS. Okay, and so those two clusters are very different. If you didn't do any of this cluster business and just averaged over everybody, here's the resulting graph. Okay, and it's actually maximum, pretty close to where Lockhart back in the 50s said the ideal range is. So it's smack dab right there, it's centered on this. But we have this big red X through it because we think that's a very misleading representation. Okay, the most that you could really say about the center region here is that that is where you cause the least displeasure amongst your consumers. If you want to maximize pleasure or liking of your coffee, you really want to serve two different coffees, one that's over here and one that's either over here or over here for these two different clusters. Okay, so that's the consumer preference testing. And oh, I forgot, I neglected to put in the snapshot of the picture, but this was published with Andrew Cotter as the lead author in the Journal of Food Science. It came out in January of this year in 2021. So Andrew did a stellar job putting this all together. Okay, so I'm getting close to the hour, and the final section here is a new chart. Okay, and so if you've been paying attention closely this whole time, you might be feeling a little bit of information overload, and that's kind of what the point of this graph is here. Oh my gosh, there's been a lot of data, all right? And so now the question is, man, how do we take all of this stuff and boil it down into something that's really helpful and at a glance useful for the coffee industry, okay? And so there's a bunch of ways we can do that. I mean, so like, and we've already done some of them. We could just take, for example, these charts. If you want to look up a certain attribute, you know, how do I want to maximize a citrus? Well, you go look at this one. You maximize it up here, right, in the top left corner. So high TDS, low extraction yield. If you want to maximize something else, okay, you know, rubber flavors, I don't know why you want to maximize that. You can look at those charts. We already have those, okay. But a different way of taking, instead of having all these different attributes in different charts, is, and this is the way we've been playing around with for a while now, one approach is, why don't we summarize where important attributes are maximized, okay? And so you can see that many of the different sensory attributes have similar trends where they're maximized in this corner or they're maximized in that corner, okay? And so here's one such representation, okay? And so here is the same format as before, total dissolved solids, extraction yield. The diagonal lines are the brew ratio. And here I have it expressed in grams of water per grams of coffee grounds. Okay. For comparison, here is the golden cup standard. I have it labeled here as the classic standard dating back to the 50s. Okay. But then in the four different corners, we have the different attributes or types of attributes that are maximized in those directions. And so we saw repeatedly that if you want to maximize sour and citrusy flavors, you want to go up here, up in this corner. Okay, and that's kind of indicated by orange. If you want to maximize the bitter and the kind of classic roasted notes, right, that's up in this corner. Sweetness, okay, and sweetness arguably should be in quotation marks here because it's not sweetness in the classic sensory science taste sense where it's caused by monosaccharides, caused by sugars. There's no perceptible or no measurable or sorry, there are no statistically significant concentrations of sugars present here causing sweetness above the sensory perception threshold. Nonetheless, our panels detect sweetness, probably because of the association or lack of masking, down in this bottom left corner of the coffee brewing control chart, and we have that labeled pink here. And then I didn't really focus on it before, but we have several suggestions that basically the tea-like and floral notes are maximized down here at low TDS and very high extraction yields. So this is one way of summarizing it, right? Now, what this doesn't do is it doesn't include any of the consumer preference testing stuff. So this really focuses on what does it taste like and where can we maximize certain attributes. And something I should really emphasize, and the one danger of a graph like this is like people think like, oh, my coffee won't be bitter if I brew it over here, because look, bitter's up in this top right corner. And so I really want to emphasize that coffee is bitter everywhere over the entire chart, but bitterness is maximized up there, okay? So the least bitter one will be down here in the opposite corner, okay? So that's one way to use this graph. So like I say, this doesn't have the consumer preference stuff, okay? So here's a more detailed approach, right? And you already saw a snapshot of this. The idea is here, let's put more of the attributes, right? So the previous graph really simplified things down to as few words as possible. Let's put more of the attributes that were statistically significant, and let's try to superimpose the hotspots for the consumer preference contours, okay? And so it's more cluttered, right? But has more information. And so same idea, we have acid, sour, and citrus are maximized up here. Astringent and viscous are astringency and viscosity, you know, in terms of the mouthfeel. Those we found those both to increase with TDS, but not with extraction yield, and that makes sense. They're just directly proportional to TDS, so that's right in the middle. And again, the bitter, roasted, burnt ash, and all this stuff is maximum up here. Sweetness is maximized down here. Black tea or tea/floral is maximized down here. And again, with the colors kind of indicated qualitatively those different regions. And then these lines right here, the solid blue line, the dashed blue line, over here and over here, those indicate where, from our consumer preference testing, where at least cluster 2, with the extremophiles, where they really preferred their coffee. And then cluster 1 was what I called, perhaps incorrectly, the sweet lovers down in that bottom left corner. So this is another way of trying to summarize all the complicated data that we put together. And in the last couple minutes here, let me just say that maybe there's a completely different way of putting the coffee brewing control chart together. And what do I mean by that? Well, this idea, which goes back to Lockhart's seminal contributions back in the 50s, the first time I saw this chart, I was confused because it's called a control chart, but it's plotted versus 2 dependent variables, okay? So when you brew some coffee, you know, the TDS and the extraction yield, you don't walk up to a machine and like dial in the TDS and the extraction yield. You change the brew ratio, you might change the brew temperature, you might change the flow rate. You change all these other actual true control parameters. You don't change those things. So this is more of an outcome graph, okay? The only independent variable on here is the brew ratio. And so very quickly, since I'm kind of out of time, we've been doing a lot of work thinking about this. And so going back to our water pulsing duty cycle business, we've been changing the duty cycle, changing the amount of time it takes. And so here's that same data, but represented in a different way. Here's the total dissolved solids versus the total brew time in a brewer, okay? And so the different colors here are different brew ratios. And the key thing here is that you can fit for a given brew ratio a nice little mathematical prediction with a little bit of noise around the curve, what the TDS is going to be as a function of brew time. And since I'm out of time, I won't go through really through the math, but there is a mathematical way of representing that. And if you do that, you could actually make a true control chart in terms of the actual independent variables. And so this is another way of formulating a coffee brewing control chart, but in terms of things that you could actually control. So here on the vertical axis is the brew ratio, and here it's grams of coffee grounds per kilogram of water. Okay, so how strong your coffee is. Here it is versus total brew time. Okay, and then superimposed on this are the outcomes, the percent extraction in the blue curves, and the total dissolved solids. So a barista or somebody could say, oh, with a certain coffee brewer, I want a TDS, and my goal is 1.5 or 1.6% and 20% extraction. Oh, here it is. That means I need about an 11-minute brew and a brew ratio of about 70 grams per kilogram. And that's how I get that right there. Okay. One caveat is that there are different access values for different grind sizes. So there's a certain grind size and for a certain overall basket. And so we're actually working on generalizing this across that. And then once you have that formulation, you can then take our sensory descriptive data, which again is a function of percent extraction TDS, and then map it onto this. And it'll have a very similar format, but it'd be a different way of reading the same stuff. Okay, so that was a lot of stuff. So I think squeezed almost 4 years of stuff into one talk. And so the very final note I'll make here is what's next, right? And so we have more papers in preparation and in review right now on this stuff, okay, based on this one project. But I just want to do a shout out that like the Coffee Science Foundation partnered with Toddy, and over the past year or so, we've also been doing cold brew research, okay? And so maybe in a couple of years, we'll have another set of papers as well, focusing on full immersion brewing and more papers on that. So I just wanted to give a little tease for that. And we have a lot of fascinating results for that. But since today's focus, again, is on drip brew coffee, I just want to end by thanking the people who did all the work. So Scott, Kenzie, and Andrew, one of the great pleasures for me is the opportunity to work with awesome students. And so I've really enjoyed working. It's a tremendous amount of work that these guys did here. Huge shout out to Jean-Xavier Guinard. Again, I'm really indebted to him for learning about consumer and sensory science under his tutelage. All the undergrads who helped. And finally, huge shout out to the Coffee Science Foundation, especially Phil and Breville for making all this possible. Without the funding to support the students, none of this would happen. So thank you so much, and happy to take questions.
Great, thank you so much, Bill. And we do have some questions here. Yeah, so here's a list of the papers Bill referred to, and Katie's just put in the chat a link to a landing page that SCA created which holds all of the academic and also lay outputs. And so you can, some of those papers are open access, so they can be accessed by anyone. Okay. So here's some questions, Bill, you ready? The first one is from Marvin Duncan. Have you explored if there is a relationship between semi-conical and cone-shaped brewers? Could some of the changes in TDS be due to flow caused by basket shape?
So no and yes. So no, we have not specifically explored semi-conical versus conical. But yes, the hypothesis that was named Marvin said, yeah, I think the main difference for the flat versus conical was the amount of time it went through. So the flat, we're keeping the amount of coffee grounds the same. So the flat geometry, it's a very thin, travel path for the water to get through. Whereas the conical, it's deeper. And so it took longer to get through, and the TDS is higher. So if your conical versus semi-conical had a different total path length, then you probably would have a difference in the final TDS, the product, for the same amount of, same brew ratio. How different it is, you'd have to do some tests and, do some experiments, measure it.
Great, thanks. Let's see. Could there be, this is about the sweetness thing, this is from Mario Fernandez. Could there be a cross-modal relationship between the mouthfeel and perceived sweetness in coffee?
That's a fancy question. Cross-modal, yeah, absolutely. I mean, so what we saw was that sweetness is maximized at low TDS, and we saw that two independent experiments. When we just looked at the final brew versus TDS and PE, and versus the fractionation as well. And so in both those, it was also inversely correlated with mouthfeel. So if you just plotted sweetness versus the perceived viscosity or thickness or mouthfeel, that was at high TDS, okay? And so there would be a negative correlation between those. So I'm not sure if that answered Mario's question, but I'm not sure why there's all these other attributes that are also, higher at high TDSs as well. And so it could be any other ones, like maybe bitterness, maybe something else. Why, that basically gets down to the fundamental question is, why is sweetness maximized at those low TDSs? The real answer is we don't know. The hypothesis, I think, is that there are different flavor molecules that your brain associates with sweetness that you can detect more readily at those lower TDSs.
Okay, let's see. Sorry. Do you have, oh, here's a question about roast degree and color for these 3 examples. I'm sure you don't have that handy, Bill. Those details are always in the papers, or maybe Bill could look it up.
I mean, it's all, oops, it's all there. I have, oh my gosh, I'm trying to, I think I might have some of them. Hold on.
I'll jump, while you look for that, I'll jump ahead to another question. Do you plan to study brew temperature with espresso style brewing? I can answer that. Coffee Science Foundation has launched a similar research program focused on espresso brewing instead of drip brewing. That's underway at actually the University of Oregon. So keep your eye peeled for news on that. We're just underway on that. Then there was a couple other questions that I can answer. Simon asked, are there plans for consumer preference testing in other markets? And someone else asked, how confident are we to extrapolate consumer preference results to other coffee consuming cultures? I'll answer that first, and then I'd love Phil's thoughts on this. First of all, yes, that's a great question, and absolutely, you can expect that preferences will be different in different coffee consuming cultures. And we'd love to learn more about that. We're actually underway in collaboration with WCR. We're looking at preferences of coffee cuppers in different geographies this year, and seeing if there are different, like if there are consistent differences with how cuppers in different cultural coffee consuming countries score coffees differently. But consumer research would be great to do. What are your thoughts on that, Phil? You live in a different culture than I do, a different coffee consuming culture. Is your intuition that those differences are measurable between these cultures?
Yeah, it was actually running through my mind when Bill was doing his presentation was just how different it would be across different nations and different consumers. And my experience is definitely there's a lot of difference between just regions in terms of their taste preferences. So I think the extreme is what you have between what I suppose North American taste preferences would be versus what the taste preferences would be in the Nordics, for argument's sake, where it's way more fruity and floral, way more perfumey and tea-like in the Nordics, and it's a lot bolder and earthier, with I suppose underpinned by a reasonable amount of bitterness would be the consumer preference for North American tastes, I would imagine, from what I've experienced anyway. So, and then you've got if you look at around Australia, it's probably somewhere a balance between the two markets, to be honest. So you've got people that appreciate bitterness, and then you've got people that really appreciate the floral and perfumey and sweeter citrusy notes of different brews.
Yeah, I agree. Bill, did you have something to add, or that information about?
I think you guys both said that very eloquently. I was just thinking, like, my undergraduate coffee class, I have, hundreds of students passing through every quarter, many international students, and like, we haven't measured it, but like just qualitatively or anecdotally, the ones from China, for example, and Korea have very different preferences for their black coffee compared to California people who grew up in California. So yeah, so we'd love to dive into that in more detail.
Okay, more technical question. Monica says, thank you for a fascinating presentation. When testing for brew temperature, did it take longer to get to the same TDS extraction yield at lower temperatures. If I ran the same brew cycle at 87 and 93 C, would the results be different?
That's excellent insight, excellent question. The short answer, yeah. Clearly, if you do the same flow rate and everything the same grind size, and just change the temperature from 87 to 93, 87 degrees will have a lower TDS at the end of the brew, just because it's a lower temperature. Things extract more slowly. So that's why when we did that set of experiments, we would alter other variables. We would like take 87 and make it a slightly finer grind size, for example, to slow it down, so that at the end you'd have, here's 93, here's 87. They'd come out with the same TDS and extraction yield. If you just, so yeah, I want to be really clear. If you change, of course, brew temperature matters a lot for the dynamics of extraction. What we're saying is something a little bit more subtle. We're saying the brew temperature doesn't matter if you fix the TDS and the extraction yield. And so those are two different statements. So, so hopefully that answered your question.
There's a question, could you clarify what responses are considered dark greens on the brewing control chart? I can answer that, Bill. Many of these references come from the World Coffee Research Sensory Lexicon, and dark green is defined this way. The aromatic commonly associated with cooked green vegetables such as spinach, kale, or green beans that may include bitter, sweet, dusty, musty, or earthy elements and may have a dark, heavy impression. The reference usually used is cooked spinach or cooked green beans. So simply, it's the smell of cooked dark green vegetables.
I believe we used canned asparagus,
Okay.
which I think that's what I've listed here. So.
Okay, if the consumer preferences in the proposed 3D brewing control chart are based on washed Central American coffee, is this also relevant for coffees of other processes in other regions?
Awesome question. I mean, so something that didn't pop up at all was blueberry, right? And so people know Giuliano, like, Ethiopian has tremendous blueberry notes, but we didn't test that, right? So we very purposely used a kind of, classic standard washed Central American style coffee to be fairly generic. But people in the specialty coffee community know there's like lots of exotic coffees out there that have crazy flavors. And the short answer is like, we'd have to do more experiments, more testing to really see how to optimize those versus brewing.
Okay, next question. Thank you for the presentation. I'd like to know more about how gas, that is oxygen or CO2, levels in the roasted coffee affect extraction. Is there more information on this?
Wow, that's a great question. So we're actually doing tests on that right now. So I put her picture up there very quickly. Jessie Liang is a food science PhD student. We're actually doing work on that exact question right now. And so stay tuned, is I guess what I would say. But in general, yes, like if you have bubbles, that whenever you have a bubble on the coffee ground, that impedes the mass transfer out of the solid into the liquid. So you got to wait for the bubble to get out of the way. So it'll definitely affect the dynamics.
Here is a question, maybe Phil could answer this. In conical versus flat bottom brewing, how would you say the final cup tastes different in general? Like say, if you had to tell it to a layman in 2 lines, who is brewing, say, in a cafe or at home?
I think you'd say the flat bottom brew is definitely, it's a bolder cup, whereas the semi-conical is, I'd say, a lighter cup would be the way I'd sort of just define those two different attributes.
Okay, here is a question from Rick Reinhardt. It might be worth noting that one of the key applications of the coffee brewing control chart was as a field tool to adjust grind size. Finer grinds generally resulted in both higher extractions and longer brew times if water volume, coffee mass, and temperature were fixed. Conversely, coarser grinds resulted in faster brew times and lower extractions. Is there room for more exploration around the relationship between grind particle size and distribution and acceptability of the resulting brew, particularly in light of the relationships between extraction and time that you uncovered?
Did you say Rick Reinhardt?
Yes.
Hi, Rick. That was a very long, detailed question. And so I think the question was, have we explored the impact of grind size on acceptability? And so we indirectly here, because to get the same TDS and the same percent extraction, we would do things like vary the grind size a little bit to change the, the flow through time and change the total brew time to nail that desired TDS and PE. So a highly relevant question then is like, how does the actual grind size affect those things? And so that's another paper that we have in process or in preparation right now. So we have tons and tons of like physical data on that exact question. As we vary the grind size, how does it change the extraction dynamics as a function of roast level as well? We're actually, just to give a teaser, what we're finding is that contrary to kind of the conventional wisdom that dark roast extracts more quickly, we're actually finding it's the opposite is true by a mile. And so there's, I think, a lot of confusion out there because actual mass transfer, how quickly molecules move out of the solid coffee grounds into the liquid, has been conflated historically with how quickly does liquid move through the porous medium. And those are two different questions. And so, and we're actually deconstructing those right now and have a paper in preparation.
I think also implicit in Rick's question, which I think is an interesting point, which is this is one of those places where the brewing control chart was invented at a time where people didn't have as much control over their brews as they do now. I mean, like a lot of these experiments were done on the Breville Precision brewer, right? Which is a home brewer, which is so adjustable in terms of not only conical and flat bottom, but also temperature and extraction time and things like that. And that's true in commercial now in home environments as well as commercial ones. And so we have the opportunity for a little bit more sophistication. Okay, let's see. Was aromatic sweetness considered as a contributor to sweet taste and flavor perception? So, and that's from Simon James. Yeah, I think
aromatic sweetness.
Yeah,
as opposed to actual taste sweetness.
Right.
So no, so like part of the like expert panel trainings, like they get together and like the panel kind of collectively decides on like what in some sample products, what attributes are best to focus on. And so that was not one that came up. And so it's the ones that we listed there that actually came up, and that's what was measured. So there are things that people might associate with sweet aromatic. Gosh, I'm not sure, like molasses flavor or brown spice flavor. I'm not sure what else, but like not as an individual distinct attribute.
Speaking of this question, the Coffee Science Foundation is really interested in this perception of sweetness thing because of how relevant it is to activities like cupping. This mystery about sweetness not being related to sucrose or other oligosaccharides, and instead being something else is a really fascinating one. And we in the Coffee Science Foundation are interested in pursuing that line of work in the future, in the near future, I would say. Is it possible to have one temperature for all types of coffee brew, origins, and roast by control all the other variables like grind size, dripper types, ratio, etc.? That's from Zichen Lee. I'm not sure I can understand that question.
Yeah, I'm not sure I can either. I mean, like, it depends what your goal is. If your goal is to have the same TDS and PE, what our work shows that like you can vary the grind size and the flow rate and all that other stuff, use the same brew water temperature and get the same TDS and PE by changing a bunch of other things. So I guess the answer is yes.
What was it that caused, this is a consumer research question, what was it that caused the extreme opposite preferences of cluster 2 to be grouped together? So you can see that those on the brewing control chart, cluster 2 is split into two. It's seemingly polar opposite subclusters or something.
here. Can you guys, oops, can you see that?
Yep. I'm sorry, cluster 1, I think he's talking about actually.
Oops, sorry, I'm shifting things around. Oh, I can't see you and me.
Yeah, we can see your screen, Bill.
Okay, there you go. All right. Yeah, so I think the question is like, why are these guys, like, why are these together, right? And so that goes back to this dendrogram business, okay? And so cluster 1 is this right here. And so we actually tested this hypothesis, maybe, because you can see we could have gone another layer down. We could have like drawn a box right here and right here, right? And so we thought, oh, maybe this cluster really likes the low extraction yield and this cluster really likes the high extraction yield. But when we did that check, we got the same saddle-shaped curve, right? So they were not differentiating based on like this group like this and this group like this. It's actually in the supplementary data for our Journal of Food Science paper that Angie wrote. And so that was a natural thing, but like natural question to ask. We checked it and it turned out there was no there. So in terms of the deeper question, like why are they liking these kind of two wildly different coffees? The reason I call them extremophiles is like, the observation is that they liked extreme values of the TDS or of the extraction yield. So it could be that it's responding favorably to a really, sharp flavor profile. Like they want something that jumps out in the mouth. So that'd be a hypothesis or as one way to interpret it. Whereas the, cluster 2, the sweet lovers, they did not like that experience, and they wanted something that was more mellow, lower in terms of strength and intensity for everything else, but like where the acceptable sweetness is maximized.
Okay, we're over time, so I'll just get to one more question, and then maybe we can wrap it up. And I know Bill would answer email questions. Hello, gentlemen. When you refer to fixing TDS and extraction, how can you expect consumers to check TDS or extraction? Also, if they have brewers with fixed temperature or flow rates. That's from Tim Sturck.
So I think I would point at Phil, right? Because the observation, most consumers don't have a TDS, digital refractometer at home, right? But this information I think would be really helpful for people running cafes and also people manufacturing coffee brewers. And so I would love to have a brewer that you just walk up to and say, click, push the button that says maximize sweetness. And then the brewer manufacturer has done all the hard work to figure out how to change the flow rate and the water pulse and duty cycle, the brew temperature to get down to that bottom left corner. That'd be amazing. Phil, can you make that happen? That'd be really cool.
I think you just articulated our next product idea, Bill.
Okay.
You can actually define the flavor that you want to enjoy from a given coffee, if that's possible, and then the brewer adjusts parameters accordingly.
Yeah, that's totally exciting to think about, and that's what this kind of research gives us the opportunity. I just want to do. So I want to. Oh yeah, Katie's just shared a link to the tool that Breville created as a sort of an intermediate step. It's meant to be consumer facing and help consumers who don't have a TDS meter or something like that craft their brew in line with this research and maximize the flavor that they're seeking to. So check that out. Bill and Phil, I just want to thank you both for your leadership and your support of this project and your commitment. It's been such a pleasure over these past 4 years working together on this, and it really has changed and just radically deepened my knowledge of coffee. So I'm just grateful to you both for everything.
It's been a pleasure to be involved with the SCA and the UC Davis team as well. So it's been a great experience from Breville's perception as well.
Great.
And so on behalf of all my colleagues and all my students, we're all tremendously appreciative. And so thank you. Thank you for making this happen.
Great. And thanks also to the audience for tuning in. Many of you stuck it out for over an hour, like an hour and a half of hardcore coffee science. That's admirable. I salute you. And with that, I wish you a wonderful Monday evening, and we'll see you at the next webinar. Thanks, everyone.
Thanks. Bye.